recipe provides an outline of one method to identify and visualize genes and isoforms that are expressed in RNA-Seq data. Given a set of raw RNA-Seq reads, the goal is to align the reads to a reference genome, estimate expression abundance levels for reference genes and isoforms, and visualize the read alignments and their expression levels. We begin by creating a subdirectory in our home directory called RNA-XP. We now want to fetch annotation files. We do this by going to the UCSC table browser. We click it and it will launch a new tab. Here we will fill in a couple parameters. First clade primates, genome human, HG19, genes and gene prediction tracks, UCSC genes, known gene, and we want GTF as the output. We also want to click the GenomeSpace button and UCSC HG19 GTF. This will automatically send the GTF annotation file to our home directory. We now want to drag and drop it into the subdirectory we created earlier. After a brief pause, the file will now be moved to the subdirectory we created earlier. It will create a dialog and tell you that it is finished. We now want to get our expression estimates from our RNA-Seq data using GenePattern. So we will go ahead and click the GenePattern logo so it will launch a new tab. In GenePattern, we will create a small pipeline which will begin with paired end RNA-Seq data in FASTQ format and run top hat and cufflinks in order to get expression estimates. We begin by setting the name of the pipeline to RNA-EXP. The first module we'll be using is top hat. This will align our reads to the reference genome. In this case, the reference genome we'll be using is UCSC HG19, so we set it as such. We also will be setting reads pair 1 and read pair 2 to prompt when run by clicking the small box. We will set library type to Illumina and GTF file to prompt when 1. As a side note, check your parameters. These are data dependent. The next module we'll use is Picard sort SAM. We connect the two modules and also set output format to BAM and change the dropdown to BAM. The next module is cufflinks. Cufflinks will generate our expression estimates. We connect the two modules, set GTF to prompt when run. Now we add FPKM tracking to GCT. This will convert the cufflinks FPKM counts to GCT, a format which GenePattern can manipulate. We set the dropdown to genes.fpkm tracking and connect the two modules. We now want to save and run the pipeline. This will bring you to the run page for the pipeline, where we will fill in parameters. The paired end RNA-Seq data we will be using is located in public, shared data, demos, analysis recipe 4, and those two files are each the left and right reads. We now drag and drop them to fill in the parameters. We now want to fill in the GTF annotation file, which we got earlier from UCSC table browser by dragging and dropping. We do this for both GTF parameters. We then hit run. This will submit the GenePattern job. As this takes a little bit of time to run, we will skip to the results. With the results, we scroll down to genes.gct and send the output to Genie. Here we hit run. Now we launch the Java app on our computer. This will open Genie. As you can see, this will allow visualization of the FPKM counts labeled by UCSC transcripts and genomic positions. Genie offers a whole host of features which you can play around with on your own. Back in the GenePattern website, we go to the home page and go back to our job results page by clicking RNA-XP on the job results tab. Here, we will want to save the BAM file to GenomeSpace, to the subdirectory we created earlier. And we will also want to save the index file to the same directory. This index file is required by IGV in order to visualize. We will also save the expression file, genes.gct, to the same directory. We now want to go back to GenomeSpace. Here, we select the BAM alignment file and launch IGV. This again will allow us to actually visualize our alignment. IGV will take a little bit of time to open and launch on your computer. IGV is a relatively memory-intensive application. Here we set the genome in the top left corner to HE19. In this case, it is already set, but if you align to a different reference genome, you can hit More and select from a whole host of other genomes. We'll now load the UCSC annotation by going to File, Load File from Server, Annotation, Genes, UCSC Genes, and then hit OK. While this loads, we will want to load the expression file from GenomeSpace. Do this by hitting the GenomeSpace tab on the top and hitting Load File from GenomeSpace, browsing to the subdirectory we saved the expression file to, selecting it, and hitting Open. This will load the FPKM counts. To take a look at the expression file, we go back to GenomeSpace and right-click on the GCT file and hit Preview. As you can see, the transcripts are labeled by UCSC names. IGV would be unable to understand what these meant if we had not loaded the UCSC annotation. Here we pick the first transcript as an example, copy it, and paste it into IGV. As you can see, the FPKM value is displayed along with a visual representation of the mapped reads. Here, the annotation is also displayed. Right-click and select Collapsed. This will give a more compact view of where the reads pile up. We can also change the range of the read count track. IGV is a rich visualization tool. I encourage you to play with it and find its useful features. Thank you for watching this recipe. Please check back on genospace.org for more to come.